Oh, good evening, everyone, and welcome to tonight's session on translational medicine with Blue Rose. Uh, tonight we'll be speaking all about neurogenetics in the clinic, and we've decided to speak primarily about a case example. We're going to speak mostly about Huntington's disease. Now, since the 1970s, molecular genetics has completely reshaped medicine. One field that emerged is this neurogenetics, and it's the study of how genes uh, are affecting the biologies or biological instructions and the code of DNA, specifically with regards to the nervous system. So that includes the brain and the spinal cord and the cell nerves and all of those things. Um, and this is both in health and disease. So among neurodegenerative disorders, conditions where nerve cells progressively get more ailments and malfunction, Huntington's disease is an inherited brain disorder that causes movement and thinking patterns to degrade and also psychiatric symptoms. Um, it's a standard model to use for neurogenetics because it's monogenic, meaning it's mainly driven by one single gene. It's also autosomally dominant. What that means is that one altered copy on one autosome or non-sex chromosome is enough to cause the disease. So one allele. And in that case, each child would have a 50% risk if the parent is a carrier. And the other reason is because Huntington's disease is what's called fully penetrant um, in the classic description of the word. So, meaning if you carry the disease range mutation and live long enough, you are expected to develop the symptoms. So that combination makes Huntington's disease uniquely powerful to understand how we go from family patterns to the gene, to the mechanisms, to eventually the therapies, which is the especially important part for clinicians. So, in that sense, Huntington's disease became sort of the proving ground for multiple genetic approaches over decades. In these webinars, we often speak about how cancer research has been a pioneering field in a lot of therapies, especially gene therapies. Now, in neurogenetics and neuropsychiatric genetics, Huntington's has been a real pioneer in this field. Because it's a single gene disorder, so one main gene explaining the condition, with predictable inheritance, the pattern across generations follows very clear rules. It lets researchers test and refine the methods, especially one particular method called gene mapping, which is figuring out roughly where a disease gene sits on the chromosome in the genome. So, and positional cloning is another method, which is finding and isolating the gene based on its location. And this is even before knowing what it does. And that's why Huntington's disease is taught as the sort of full pipeline disease, because if you know the full pipeline of Huntington's disease, you can apply that to most other neurogenetic diseases as well. Clinically, Huntington's disease, quite a tongue twister, uh, presents with a progressive phenotype, meaning that the observable features of the disease um, usually begins in adulthood or later in life, and also worsen over time. So, and classic symptoms include chorea, which is involuntary movements, cognitive decline, so that's the worsening of memory, planning, thinking, that type of thing, and then psychiatric disturbances. This includes mood and behavior, and sometimes even psychosis-related symptoms. And death often occurs within 10 to 15 years after the symptom onset. So, in genetics, complete dominance means that having one mutant copy of the gene produces essentially the same full disease pattern. So, in genetic terms, so the heterozygotes, meaning they have one normal allele and one mutant allele, and the homozygotes, which is very rare in Huntington's disease, but meaning they have both mutant alleles, are the same. They both result in Huntington's disease. There's not really a clinical difference between the two. What makes Huntington's disease a clinically heavy combination um, of predictability. Oh, yeah, that's what makes Huntington's a clinically heavy combination of predictability and timing. Right. So families often notice this multi-generational pattern, meaning multiple generations show similar symptoms, and yet the onset is usually later in life. So this makes it very difficult because people can spend decades living with uncertainty about whether they will develop the symptoms, and also whether they may pass it on to future generations. So the risk of the risk of sending that to the future generations or to children. So that's where genetics becomes not just science, but also ethics and mental health and medicine, of course. So let's talk a little bit about the history, the whole mapping of the Huntington's disease. So the breakthrough concept um, was that you could map this disease using DNA markers. Now, a DNA marker is measurable DNA differences that vary between people. Um, and you can do this without actually knowing what the gene does yet. So this process is called reverse genetics. That's if you don't know what the gene does yet. So you start with the inheritance patterns in families and track markers that travel with the disease. Then you locate the gene, and only later you can, after you have the gene through all sorts of silencing and inducing methods, you can figure out what it actually does. If you were here for last week's webinar, you would have heard about the forensic biology one. You would have heard about RFLPs, which stands for restriction fragment length polymorphisms. And these are DNA differences that change where restriction enzymes, which is a molecular scissor or a molecular enzyme that cuts DNA at specific sequences, um, and this produces fragments of different lengths, so which can be visualized as a through a method called Southern blot, which is a lab method that just separates DNA fragments by size and detects specific fragments using probes. So, in simple terms, the RFLP are different DNA markers or cutting patterns, and you can, in a visualization method, you can see them and read them out in a lab report. Um, so using these molecular markers and also using something called linkage studies, um, we were able to discover where the genes are. So what is linkage study? Linkage study is just a test that asks whether a marker and a disease are inherited together. And linkage is what that means. So um, and a linkage study is only as powerful as the pedigree, a pedigree being the family or the family tree within which the disease resides. So if you have a very, very large pedigree, you have a large amount of information, and this increases the statistical power, meaning you can be much more confident that a marker is truly tracking with the disease rather than matching by chance. But in theory, the concept is if you constantly see a disease and a certain, let's say, let's say gene, for simplicity's sake, are inherited together, this suggests that they're in linkage, but there is more nuance to that, and we'll go into more nuance in that in a second. So a turning point historically was finding a marker whose inheritance tracked with Huntington's disease in a very large pedigree. Um, and they found that something called the G8 marker produced different haplotypes. Haplotypes used to confuse me a lot when I was still doing my undergrad. So a haplotype is just a package of DNA variants that are inherited together in this on and usually on the same chromosome. In fact, some definitions would imply that it has to be on the same chromosome. Um, so when one haplotype is consistently inherited along with the disease, it strongly suggests or strongly implies that a marker sits near the disease, in this case, Huntington's. So in that way, if you know that things are close to a certain gene and that these genes are inherited together, you can start narrowing down which chromosome is going to be the culprit. So because this DNA tends to be inherited together, and so what you can do once you have the linkage established, the next question is, okay, which specific chromosome carries the marker? And there you use a method called somatic cell hybrid mapping. And so this uses lab-grown cells that contain different combinations of human chromosomes. And if the marker signal appears only when a specific chromosome is present, you can assign the marker to that chromosome using this approach. So the Huntington's disease marker was linked to I think, correct me if I'm wrong, by all means, but I believe it was linked to chromosome 4. If you think of the human karyotype, a karyotype is all of the chromosomes of a person laid out, labeled from 1 to 23. And this after they assigned chromosome 4 to the one linked to Huntington's disease, this launched the um, modern methods or modern start of the positional cloning in human disease era. So great, once you have the chromosome, and apologies if I speak a lot, there's just a lot of information in this in tonight's webinar crammed in. So after you've identified the chromosome, you narrow it down to a candidate region or a specific locus. Um, and the nomenclature that they use uh, usually goes from the short arm. So in Huntington's disease specifically, so it's on chromosome 4, so it would be 4p, that's the short arm of chromosome 4. And then there would be a number after that. Um, and that number would be the band location. So I think Huntington's is 4p16.3. Um, so essentially, it's just saying that on this chromosome, uh, on, on the short arm, this in this band region or band location is where you'll find the Huntington's area. So, and this researchers, but then what they do is they build sections called contigs, which are overlapping clone DNA fragments, which are also used in sequencing, which we'll talk about at some other time. Um, and so what they do then is they try and cover the whole region like this with these different contigs, and then they systematically search for the gene. So they use they use something called exon tapping, which is a method that helps pull out exons, which are the coding parts of genes, um, then that end up making messenger RNA. So so and the messenger RNA is kind of like the blueprint. So if you think about something called the central dogma, DNA makes messenger RNA, which makes protein. So from these large DNA fragments, those exons are then used to find which genes are being expressed. Um, in other words, it finds what we call candidate genes or candidate gene messages if you're looking for the RNA. And one key transcript in Huntington's disease, the IT15, stood out because it affected individuals, um, or it essentially caused what we now know to be the CAG repeat. So CAG refers to nucleotides, cytosine, adenine, and guanine. And so these three nucleotides in in exon 1, so the first exon that they got from that actually codes for something, repeated. Um, and if this repeat happened too many times and crossed a certain threshold, they found that yes, this was linked to Huntington's disease. So we learned that, oh, okay, so Huntington's disease is not only just a region, but also specifically in this region, there's this three nucleotides, the CAG nucleotides that repeat, and the more they repeat, the more likely it becomes that Huntington, or it is that Huntington's will become expressed. And we'll go into a little bit more nuance there in a second as well. So in 1993, the Huntington's gene was identified. It's called HTT, which encodes for the protein called Huntington. And Huntington's disease is one of uh, the trinucleotide repeat diseases. That's meaning that the mutation is expanded repeat regions of these three-letter DNA codes. In this case, the CAG codes for an amino acid called glutamine. So these expanded and repeated CAG uh, areas uh, produces something called polyglutamine or polyQ, and because that happens, it means essentially that there's just too many glutamines in a polypeptide chain or in a protein, and that means that it folds wrong, and this can cause toxicity. So clinically, testing usually means measuring the amount of CPG. Sorry, not CPG. That's something else. My mind is still on last week's methylation. The CAG repeats. Um, Now the main lab method to do this is PCR, as it usually which is polymerase chain reaction, which is a technique that that couples a targeted DNA region um, uh, with primers, and then these primers in a thermocycler essentially amplifies that region through amplicons, and then you have millions of copies that you can use for testing. So, and the testing scenarios include everything from symptomatic confirmation. So it confirms whether a person has Huntington's disease. You can do pre-symptomatic testing. So that's testing an adult who is at risk but doesn't have symptoms yet. And it also includes something that we spoke about in one of our very first webinars, which is prenatal testing. So testing an embryo or a fetus. But the hard part isn't actually the PCR testing, it's or the interpretation, but then the counseling. So that's what you would need a genetic counselor for, because there's a lot of psychology and ethics involved in the whole process as well. So, just to give you some exact numbers on Huntington's disease. So, if you've done a test, if you find that there are less than 26 repeats, this is normally this is still within the normal range. You don't expect Huntington's disease to show up if you see that. If you see more than 26, but still less than 35, they would start calling that intermediate alleles. So it's still not expected to cause the disease in that specific individual, but there is a little bit of a generational risk in some contexts. And then once you start going over 35 repeats, so say from 35 to 40, you already start seeing the disease sometimes showing up. So that would be penetrance. So if the markers are there, but it's only being expressed some of the time, that's its penetrance. Um, and then as soon as you reach a point where there's over 40 repeated markers, then the risk of presenting with Huntington's disease becomes very, very high, almost 100%. Um, and remember, this is an autosomal dominant disease. So because of that, if you have over 40 repeats in any one of your alleles, but let's say you just have it in one, then there's a 50% chance that you are going to pass that on to a child as well. So yeah, that's just the mechanics and also of the actual nucleotide number and how the number of repeats relate to the onset and threshold of Huntington's disease. So clinical guidelines often emphasizes quite a few things. So the first thing would be informed consent, as usual. So this just means that the patient when they do a test for Huntington's disease now, this just means that they understand the benefits and risks and consequences of knowing the information. Confidentiality, of course. So this means that the results have to be protected because they affect relationships, employment, insurance, mental health. Um, in fact, you actually have to do a neurological evaluation. So um, that's that's assessing signs of nervous system dysfunction, and a psychiatric evaluation, which is a mental health screening. So you have to see if the person who does the test needs to actually go see a psychiatrist to see whether they're at risk for depression or suicidality. Um, and above all, that when the person does the test through all of these evaluations, the results need to be delivered in a structured report, but that comes with support. So so that would be where a genetic counselor or a psychologist or a psychiatrist plays a massive role in actually delivering the very, very heavy results of Huntington's disease to a patient, but any degenerative disorder or neuropsychiatric disorder as well. Um, and also, just on that ethics, a little interesting side bit is that that testing on minors is strongly, strongly discouraged and even essentially illegal in some places. Um, and except there's an exception, as there always is in something called juvenile, or if when you expect something called juvenile Huntington's disease, which is a rare early onset form of the disease. But the guiding principle ethically is autonomy. So the person's right to decide whether they uh, want to know when they are old enough. So at 18 years old, they can choose whether they want to know the results or not. So Okay, so that's some of the ethics. So let's just talk a little bit about um, let's talk about the mechanics of Huntington's disease. So Huntington's disease is a mutation described as a gain of function mutation, meaning that the mutant protein doesn't just lose its normal function; it actually gains a harmful property, right? This expanded polyQ tract makes Huntington's disease. This is the expanded amino acid tract, so this makes it prone to misfolding. In other words, folding the protein folds in different all sorts of different shapes, and these misfolded Huntingtons form aggregates, which are clumps of protein, and those aggregates can sequester regulatory molecules, meaning they essentially trap and tie up proteins and cells that are needed for normal control, causing a widespread systematic dysfunction. So it's another example of a complex network-based disease. Now, Huntington's interacts with many proteins. Um, so the mutant form disrupts multiple systems from transcription. In other words, how genes turned on into RNA, messenger RNA, how proteins fold and degrade, and in other words, the quality control. If you think about the brain, Huntington's obviously affects the synapses as well, so communication between junctions of the neurons, and it even affects mitochondria, which, as every biology student or anyone who's ever done biology in their life knows, is of course the powerhouse of the cell. And let's talk. So let's talk about a little bit um, of detail there. So, and I see some people. So you're welcome to ask questions at any time. Welcome to interrupt. Um, and you're also welcome to email me at time. I didn't say it in the beginning, but please, by all means, interrupt and ask questions. So let's talk a little bit about those disruption mechanisms in detail. Uh, One key mechanism is the transcriptional disruption, meaning genes are less able to turn on normally. So mutant Huntington interferes with uh, enzyme called histone acetyltransferase, which I love the little acronym for it. It's called HAT, and enzymes that which and they're essentially just enzymes that add acetyl groups to histones. So and histones are the proteins that DNA wraps around. So acetylation usually keeps these chromatins more open, allowing for transcription. But if the hats are blocked or the HATs are blocked, then you get what's called histone hypoacetylation, which are too few acetyl marks, and this promotes heterochromatin. So that's the regions of DNA that's really tightly packed, and these closed chromatin regions reduces something called the promoter accessibility. So if you remember last time we spoke about promoters as well, these are the genes that essentially, or the regions of a gene that essentially switch on transcription. Um, and yeah, because these are impacted, that of course reduces transcription initiation and the start of the messenger RNA production. So that's how transcription disruption happens in Huntington's disease. The other thing that I mentioned was protein folding. So when gene regulation is disrupted, the cell's folding helpers suffer. So chaperone proteins are proteins that help other proteins fold correctly. They chaperone them. But if a chaperone system is impaired or misfolded, the proteins accumulate. So this results in a failure of proteostasis. Proteostasis is the cell's ability to maintain sort of a healthy um, protein environment. That's the protein folding. The other part that I mentioned was impaired degradation, right? So the cell always tries and clears damaged proteins using a different protein called ubiquitin, because you find it everywhere. It's ubiquitous, and it's a small tag that marks proteins for disposal. These tag proteins are usually destroyed uh, by the proteasome, which is literally a cellular shredder complex. It destroys other proteins. But in Huntington's disease, the proteasome clearance becomes ineffective. Um, and and it's because of these aggregates again. So the toxic material persists, and that chronic stress caused by these being around can push neurons towards apoptosis. So the neurons are actually dying. Um, in apoptosis, as you know, is called cell death. Is another word for cell death. Um, and then the second last function that I mentioned there was synaptic dysfunction. So this is actually quite an important one clinically, um, because it refers to the problems at the synapses, so the places where the neuron to neuron communication happens. Um, and this can appear, this is one of the first signs that you see that appears before neuron loss. Um, in transgenic models, so a transgenic model would be usually an animal model that's engineered to carry, if it's a human transgene, it was engineered to carry a human disease gene. So in these transgenic models, synaptic vesicles, which are tiny packets that carry neurotransmitters, um, have been shown in many, many studies to be abnormal, and signally impaired. So one implicated molecule in this whole system is one called RAB 11, I believe, which is also it's a little bit different from a chaperone protein. It's called a trafficking protein. So it's involved in recycling neurotransmitter packets or vesicles. But in these lab mice, increasing the RAB uh, activity actually helped restore vesicle function, which suggests that early sort of function first targets may exist before irreversible degeneration. And then I also mentioned the powerhouse, but everyone's favorite powerhouse of the cell, the mitochondria. Uh, as you may logically think, that energy failure can always play a role. So the mitochondria generate cellular energy in the form of ATP or adenosine triphosphate, which is the all cells, or biological material is ATP. It's its energy, right? So uh, in the case of mutant Huntington, this binds to the mitochondrial membranes and impairs what's called the electron transport chain, which is this core energy making process. If you have less ATP, this actually also raises the reactive oxygen species, or the ROS, which are highly reactive molecules that can damage proteins as well, and lipids and DNA. And then finally, the mitochondria actually need to move to the synapses, but when the damage becomes so high, this trafficking protein becomes so impaired that it means that the synapses become completely energy starved, um, completely worsening the functional decline, and eventually leading to the complete deteriorations. So we spoke a little bit about transgenic mice there. Um, and I actually think it's important to speak about some of the other uh, animal models, and why it is important. So human neurodegenerative diseases is very hard to study. So animal models let you control the environment, observe pre-symptomatic stages before symptoms appear, and also test for interventions at very specific time points, which is something that you can't really do cleanly in humans. So this is why mice models, or transgenic mice models carrying the HTT gene, the human Huntington's disease gene, with different varying lengths, um, are very good for studying. And so what was actually found from studying mice and these where these repeat regions were longer, is that the longer the region is, the earlier the abnormality is, right? So that's something that we found from mice models, and especially in the striatum, which is a brain region involved in movement and control and habit circuitry. So this is actually the striatum is the region that shows the most vulnerability to increased lengthy repeat regions, that CHE region of Huntington's disease, which also maps to the clinical pattern seen in humans. So what we found in transgenic animals or animal models, we also saw clinically in humans. Um, But one hopeful point of principle is that also through animal models, we were able to study something called an inducible expression model. So that what that means is that the mutant gene could be turned on or off, usually through some sort of drug. You also get a version of that where they can turn it on or off using light, which is quite interesting. We'll talk about that at some point as well. So when a mutant HTT expression is switched off, to say with drug, um, after the symptoms already began, right? What they saw in animal models is that aggregates actually started clearing up, and motor symptoms started improving again. And so the clinical implication of this is that in theory, this is not something that can be done or is done in practice just yet. But in theory, the earlier the disease is picked up and the reversible gene silenced or turned on, um, the more likely it is that the disease, Huntington's disease, is actually more reversible than we initially thought, which is very promising for the future. And that might so, but of course, you also get large animal studies because there's a lot of differences between mice. So mice are powerful, but limited. They have short lifespans and short brains, not short brains, small brains. Um, where something like large animal models, like sheep, can much better mirror long-term diseases and allow for imaging that's a lot more comparable to humans. So you can, for instance, you can do MRI, which is a structural brain imaging on a sheep, and you can do PET scans on a sheep. Um, and so that matters a lot for translational work, is bridging the gap from the lab to the clinic, which is often why medical studies use sheep or pigs, so larger animal models. And the therapy strategies that they map onto these mechanisms usually have to support all of the pathways that we spoke about throughout this webinar. So that includes neuron survival, that includes improving protein quality control, or reducing the amount of mutant Huntington, so that you don't get these aggregated protein clumps. Um, in other words, you're trying to sort of early on stop the upstream driver of this cascade of damage. And one approach is trophic support. Trophic, anything that's trophic refers to something that tends to have a cascading effect. So BDNF is brain-derived neurotrophic factor, um, and it's a survival and function signal for neurons. So especially this becomes relevant in the striatal structure and striatal health. So we use something called mesenchymal stem cells, which are adult stem cells that have been engineered as delivery vehicles um, in animal models. And when these engineered cells have overexpressed BDNF, um, it tends to improve neuron survival and behavior, which is very experimental. Um, and as far as I know, hasn't been done in humans, but in animal models, it illustrates that if you provide support to these vulnerable structures, it can lead to improvements. So, and this is essentially a classic case of drug discovery. So what you do from there is you take high throughput screening, which means testing huge numbers of compounds to quickly find candidates that shift a um, target pathway from negative to positive, of course. So yeah, the goal is to boost the protective factors, to try and ultimately stop these folding patterns that you see in Huntington's disease. So that can be via transcription factors that regulate chaperones or directly, by reducing the aggregation. And even a modest shift in protective gene expression can actually have measurable effects in models. Um, and that's only the start of it. So in modern times we actually use gene silencing. So that's if you take a mutant Huntington that drives the cascade and lowering its level directly through gene silencing, and essentially reducing the gene output. What you then get is that two post-transcription strategies can essentially target um, the onset of this disease and the therapy of this disease. So the one method that we use is called antisense oligonucleotides, or ASOs, which are these short sort of DNA-like molecules um, that bind to RNA, and the other one is RNAi, which is, I think it stands for RNA interference. Actually, I know it stands for RNA interference. And so this is a natural system that uses small RNAs to guide RNA destruction or suppression. And both of these aim to reduce mutant Huntington's disease messenger RNA. It's very similar to the concept that I spoke about 2 weeks ago with microRNA that also binds to messenger RNA, and instead of silencing it, it actually degrades it. RNAi just silences it, but it does ultimately do the same thing. It makes less messenger RNA, which means less of the negative protein is transcribed. Um, we can go into a little bit more detail on that SAO. So essentially, what they do is they form this DNA RNA hybrid, and then they recruit an enzyme called RNase H, and that cuts the RNA strand. And because of that, that reduces protein production, and um, and interestingly enough, that same RNA, once it's cut one, that same ASO, once it's cut one RNA, it can actually go and bind to a different messenger RNA. So one can cut multiple, similarly to how one microRNA is capable of binding to multiple messenger RNAs. One ASO is literally capable of cutting multiple messenger RNAs. Um, the problem is, even though theoretically we know it does this, it's the delivery of the drug that becomes difficult therapeutically. So in theory, if you have an ASO drug, you can deliver that into the CSF, the cerebral spinal fluid, or just the watery regions that surround the brain and the spinal cord. But it's easier said than done. What we are starting to do, on the other hand, is genetic editing. So that means changing the DNA itself directly, rather than just reducing the RNA through messenger RNA, microRNA or RNAi or ASO methods. We're starting to use the very popular method called CRISPR-Cas9, which is widely starting to be used in editing systems for all sorts of purposes. And all that it does, really, it takes a guide RNA that recruits a cutting enzyme. In the case of CRISPR-Cas9, it's the Cas9 enzyme. And then what it does is it cuts a DNA sequence. And so for a dominant disorder like Huntington's disease, the elegant goal would be to silence or cut out the mutant allele while sparing the other one. uh, so it's that, and that process is called allele specific targeting. Then, so essentially it means that you're using a very small sequence, um, often single nucleotides, in fact, to try and only target the regions that are faulty. In the case of Huntington's disease, it would be the repeat region of the CAG region. The only problem with CRISPR is that it can only be recruited next to a region called the PAM region. And so, yeah, it becomes a problem because you as accurate and neat as CRISPR is, it doesn't actually cut exactly where we want to cut, because it does need to still actually be adjacent to the PAM region. So it is useful in that it can cut very, very close to the right amount, but can't actually selectively cut the specific region. But that's where other gene editing technologies is becoming very, very useful. Something like Tiger, which we spoke about a month ago, um, is potentially usable for this application. So, okay, so let's step away from the gene editing for a second. So Huntington's disease is part of, like I said earlier, of the trinucleotide repeat disorder, many of which are these polyQ diseases, this repetition of the amino acid. Um, And this repeat of the CAG region causes polyglutamine, which causes toxicity. Um, and the more of these repeats there are, the likelier the chance of earlier onset becomes, as we saw through the animal models. This, by the way, has a genetic term as well. It's called genetic anticipation. So, and it's not just earlier onset, it's usually more severe as well. So, and this actually strongly, come to think of it, links up to paternal inheritance as well, meaning the expression um, happens more frequently when the mutant allele passes through the father. So, which is an interesting, and I don't know why, but it is more common to be passed on through the father, which is interesting because um, it's an autosomal sequence, so it's not linked to the sex chromosome. So it's interesting that it's passed on more readily through the father. But I don't know the mechanisms. I'm very open to being told what they are. So a shared modern theme, however, with these misfolded proteins is that they can spread between cells. So one route is passive. When the neuron dies, it aggregates, and this can be released and taken up by the neighbors. And another route is active through exocytosis. So that's when cells release materials outwards into vesicles, followed by endocytosis, which is when other cells, usually neighboring cells, take up those cells. And so this is how neurons communicate. So synapses through exocytosis send out neurotransmitters in these vesicles, and on the other side of the synapse, they take it up again. And so this is why there's a big implication for neural tissue um, in the spread of this thing, is just because inherently of the way that the tissue functions, it is sharing these packages, and that is why Huntington's tend to affect the brain regions a little bit more. So I see we are starting to run out of time. I did have a few other things I wanted to say as well, but I think we did explore most of the big things. So uh, if Huntington's taught anything in terms of medicine, it's that genetics can be operationalized. And you can look at data from families to single genes to mechanisms to pathways, all of these to inform on which therapies can be done and have been done, either in humans or animals, and it opens the door to clinically testing, um, which is often more straightforward than we were thought, but ethically very heavy. And then mechanistically, the mutant Huntington we know disrupts multiple core systems. So it unfortunately isn't necessarily just a case of figuring out the symptoms and then relating that to a single physiological, say, organ system, but multiple gene systems, even though it is regulated by one gene repeat. That one gene repeat has effects on other gene regulations, on protein homeostasis, on synaptic communication, and as we said with the mitochondria, cellular energy. Um, but the therapy frontier is increasingly focused on reducing mutant huntingtin, and to try and sort of target this upstream effect that it has early on, rather than trying to treat the symptoms after it may be too late already. And so, with that, unless we have any last minute questions, I bid you adieu. And I hope this Huntington's disease case study has been interesting, and that it was a good example to use for neurogenetics. And if you're interested in hearing more about neurogenetics, but perhaps less specifically Huntington's disease, you can also let me know, and we can do a more general overview. Uh, but until then, good night, and I hope you enjoyed. Thank you.